Odin is a patent-free research network for industry and researchers at Aarhus University. We aim to accelerate drug discovery through open innovation in science. The network is sponsored by the Novo Nordisk Foundation. P2P CPP, optimization of cell-penetrating peptides to enable therapeutics with intracellular targets, is one of our funded projects.
Our project here was initiated by Stipe Therapeutics, and they have asked us to develop assays to understand what is the physiological effect of these cell-penetrating peptides.
Stipe Therapeutics is looking to develop drugs for immuno-oncology applications and also for treating autoimmune diseases. So our lead molecule is a molecule designed to enhance the immune response to cancer. It relies on a cell-penetrating peptide in order to deliver the active part of the molecule into the cell. Still isn't quite a lot known about how these molecules deliver their cargoes into cells. And that really led directly to the idea for the Odin project. How can we understand the mechanism of entry of CPPs into cells, and how can we do that in a manner that is both optimized for delivery and at the same time minimizes any other effects that these cell-penetrating peptides might have.
A huge problem in developing pharmaceuticals is that you can't target things inside the cell very easily. The cell is protected by a bilayer membrane, and a lot of pathways that are involved in diseases are inside the cell. In order to pass this outer plasma membrane, you need something that has the very right physicochemical properties. The aspect that we are working with is a peptide called TAT, and that's developed from the HIV TAT protein. And it's not understood why, but this is able to penetrate the membrane. But with anything where you are making a hole or penetrating, you also risk actually destroying the cell or affecting it negatively. So this has been seen also to have larger physiological effects. We want to be able to predict what will the effect be when you use this systemically. And we have developed some novel assays where the hope is that we can actually predict what it will be. And we're also hoping that this will tell us what are the physicochemical properties that determine how big a hole is this making, or how much of an effect does it have, and that this can also be used then to predict just from a sequence down the line what will be the effect in the body of a potential novel drug. And we believe in general that any pharmaceutical company that wants to target something intracellular with biologics could be interested in having an optimized strategy to get something into the cell. So we hope that we can at least help on the way of developing a method where you can predict an efficient and very safe way of getting cell-penetrating peptide-based drugs into cells.
For us, one of our aspirations for the Odin project is that we will get new information on how to design better cell-penetrating peptides for this generation of immuno-oncology projects, but also for future projects that are at an earlier stage within the Stipe R&D portfolio.
All of our projects will share their data and results openly with the public. To find out how you can access them or join the Odin network, please contact us directly or visit our website.